Hello friends, welcome back in this video session of medicinal chemistry. So today we will be discussing an important concept in the medicinal chemistry, that is drug metabolism. Drug metabolism is also referred as a biotransformation. So before starting the biotransformation or the drug metabolism, we know our human body is so beautifully engineered. It has many organs in the body and it has many functions, and we have several enzymes in our body. There is a lot of biochemistry involved in the body, and we know our human body is exposed to many exogenous or the foreign substances, and the body has its own mechanism to tackle these xenobiotics. So xenobiotics are nothing but these are the non-essential foreign compounds. So any compound which is not required to the body and it is given externally, right, it is introduced from the external source into the body, we refer this as a xenobiotic. And throughout our life we consume varieties of drugs. So these drugs are also considered as a foreign substances. They are not essential to our body, but for certain actions, for certain disease treatment, we take those medicines or the drugs. Then we have natural toxins from fruits and vegetables what we eat, right. So those are also considered as the foreign substances, and we usually smoke or drink, and that will also have the toxic substances. So body has its own mechanism to tackle or handle these kinds of substances. Okay. So the major organ which is responsible for handling the xenobiotics or the drugs is the liver. So liver is the main site of metabolism. Okay. So this we can refer it as it is a primary site of metabolism, where the drugs and the xenobiotics are the non-essential foreign compounds or substances which are getting metabolized, which are getting biotransformed into some other chemical forms. Other than the liver, there are of course the other organs which are also responsible to some extent for the metabolizing these kind of substances. So these are the lungs, kidney, intestine and plasma. So most of these xenobiotics, they undergo a biotransformation, as I told you, in the liver and extrahepatic tissues, and are excreted as hydrophilic metabolites. So usually when we perform the biotransformation in the body, what happens? The compounds or the xenobiotics, they will be biotransformed or they will be converted into a chemical form which has more polar functionalities, and because of that they are hydrophilic in nature, and because they are hydrophilic in nature, they can be excreted easily from our body. Okay. So basic idea behind the biotransformation is we need to have the excretion of those substances from the body. For excretion we require hydrophilic conversion. Okay. Sometimes these metabolism processes, it causes the mutagenicity, toxicity, cytotoxicity and carcinogenicity, and therefore it is very important we need to understand the biotransformation, especially the chemistry involved behind these biotransformation of xenobiotics and the drugs. So for most of the drugs we require a metabolism. There are few exceptions where the drugs doesn't require any metabolism, it will be directly excreted from the body after its pharmacological action, but most of the drugs they require the metabolism, right. And you can imagine, if the drug is not requiring the metabolism, it will be excreted, but if it is requiring metabolism and if it is not getting metabolized, then it will have several problems, it will have several issues. So the first issue is it won't show its pharmacological action, because sometimes the drugs are not active, but it will be converted into some chemical form which we consider it as a metabolites, and the metabolites are active, they are showing pharmacological action. So unless and until you are converting the inactive drug to the active drug or the active metabolite, it will not have a pharmacological action, right. So there is no use of consuming such type of drugs which are not getting metabolized, right? If the metabolism is not happening in the body, this will not show pharmacological action. And another problem can happen, it won't get excreted, because most of the drugs we know they are lipophilic in nature, because they have to cross the lipid or lipoidal barrier, right, and they have to reach to the receptor site. So once they show their action, they need to be excreted. So for excretion they should be hydrophilic. Okay, if they are not hydrophilic, what will happen? During the renal reabsorption, in the renal tubule they will get reabsorbed in the body, and they will accumulate in the body, and they will cause the toxicity, okay, due to the accumulation. So these are the several problems which we can encounter if we have the problem with the liver, because liver is a major site where the drugs are getting metabolized, and most of the drugs require metabolism or the biotransformation, and that is why we need to understand the drug metabolism or the biotransformation. Now the drug metabolism, because what considerations we have discussed, it plays a very important role in the elimination of drug and xenobiotics from the body. So drug metabolism or biotransformation, we can define it is a chemical alteration of drugs or xenobiotics by a biological system in the body. So usually we convert our drugs into a hydrophilic substances in a hydrophilic chemical forms, which can be excreted from the body, and certain drugs require bioactivation, it requires to be converted into its metabolite, and those metabolites will be active. Okay, so it is needed to reduce the non-polar, that is lipophilic drugs, to a polar, that is hydrophilic form, so that they cannot be reabsorbed in the renal tubule and that will be excreted. Some of the drugs which are already hydrophilic in nature, they does not require much alteration in their chemical form, and they will be easily excreted in their unchanged form. So we have an example of streptomycin, neostigmine, which does not require the biotransformation. Okay, so they are already hydrophilic in nature, so they have ability, without converting into a other polar form, they will be getting easily excreted because they are already hydrophilic in nature. Okay, so what are the different sites of drug metabolism? So as I told you, the primary site for drug metabolism is liver. So that's why the people who are suffering with the liver diseases, they have a problem of toxicity, they have a problem of drug metabolism, and we need to consider those things in the liver patients. So it is a main site for metabolization and detoxification of many endogenous and exogenous compounds. Okay, so liver is the main site because it is highly perfused organ, and it is also rich in many drug metabolizing enzymes. So that is the reason this is a primary site for metabolizing the drugs. Drugs which are absorbed in the GIT reaches systemic circulation, and then it goes to liver to get metabolized. So this is what we refer it as the first pass effect or the first pass metabolism. Okay, so what happens in the first pass metabolism or first pass effect, the drugs are getting absorbed through the GI tract, and then it is going into a systemic or blood circulation, and then it is going to the liver, and then it gets metabolized. So this is basically an first pass effect or the first pass metabolism for the drugs which are getting absorbed through the GIT. There are of course other sites of drug metabolism other than the liver, and they are referred as a secondary site of drug metabolism. So what these secondary sites or organs are? So kidney is one of the secondary site of drug metabolism, where the proximal tubule is the main part of the kidney which is responsible for excretion of these kinds of drugs. Then of course we have lung, which is basically having a type 2 cells, then testis which have the Sertoli cells, then skin having epithelial cells, then intestine, plasma, nervous tissues of brain. So these are the sites which we can refer it as a secondary sites, because the organs which are considered as the secondary site or other than the liver, they have very less amount of metabolizing enzymes. Even the varieties of drug metabolizing enzymes are also in less amount as compared to that of the enzymes present in the liver. So, that is why these are called as secondary site of drug metabolism, and this we can also referred as the extrahepatic metabolism. So, the metabolism which is not happening in the liver, which is happening in the other sites or organs of the body, that we refer it as a extrahepatic metabolism, and this is of lesser importance because of lower levels of metabolizing enzyme present in these tissues. So, what is the purpose of drug metabolism? Okay. So, in which case or in which kind of examples we require the drug metabolism? So, first case is the inactivation of drug. So, we need to convert the active drug into its inactive form, okay, and we can excrete that out of the body. For example, we have an phenobarbital. So, phenobarbital is the active drug. So, we are giving phenobarbital as such, and this is active drug, right, it is active form, but when it undergo a biotransformation or when it undergo a metabolism, it will be converted into a hydroxyphenobarbital. So, we can see here the hydroxyl group is introduced to this particular benzene ring. So, because of the hydroxyl group, we know hydroxyl group is polar in nature. So, we are converting the non-polar phenobarbital to the hydroxyphenobarbital. Phenobarbital has the action, it is an active form, and hydroxyphenobarbital is an inactive form. So, we are converting active form to the inactive form, we are converting non-polar form to the polar form or lipophilic form to the hydrophilic form, and these polar kind of substances can be easily eliminated from the body. It will be not getting reabsorbed in the renal tubule and excretion is feasible. Okay. So, we have many examples of drugs which requires the inactivation, right. So, once they show their pharmacological action, they will be inactivated, they will be converted into a hydrophilic form, inactive form, and then it will be excreted. We have an example of ibuprofen, paracetamol, chloramphenicol. So, these are the examples of drugs which requires inactivation, and this is one of the purpose of the drug metabolism. Then second is active metabolite from the active drug. Okay. So, in this case the active metabolite is converted to active drug, and this is again for the purpose of excretion. So, here you can take a very classical example of this type, that is a diazepam. So, diazepam is a active form. Similarly, the oxazepam is also active form. So, both forms are active, both will have the pharmacological action, but here we need to keep in mind the main difference between these two is here we can observe there is a hydroxyl group, right. So, rest of the structure is almost same, and then there is one more difference here, that is NH. So, the hydrogen is substituted here in place of this CH3, right. So, hydrogen and hydroxyl group is making diazepam to oxazepam, and oxazepam is more hydrophilic because of the introduction of these polar groups, right. Both forms are active, but diazepam cannot be excreted because it has a lipoidal nature, and hence it will not get excreted, it will be in fact reabsorbed in the renal tubule, and what will happen, it will show its action for a prolonged time and it will not be eliminated. So, that will create a toxicity, it will get accumulated, right. So, which we do not want. So, what we require, we biotransform, we do chemical alteration in the structure of diazepam by metabolizing enzyme and convert it into oxazepam. So, both have the activity, both have pharmacological action, but this oxazepam can be excreted because it is hydrophilic in nature. Similarly, we can take an example of amitriptyline. So, amitriptyline is the active drug. So, by mistake it is written inactive. So, this is active drug and its it will be converted into a nortriptyline. It will be converted into a nortriptyline. So this is, both are having an active form, and you can see this NH is introduced here. Okay. So this will be more hydrophilic in nature, and then it will be easily getting excreted. Okay. Then the third type is the activation of inactive drugs. So in the first type we have seen the drugs were active and it were converted to its inactive form. Second type we have seen the drug was active and its metabolite was also active. So there is a summative kind of pharmacological action what we can observe in the second type. In the third type, activation of inactive drug. So what we refer it in a pharmaceutics point of view, these are the prodrugs. Okay. So they themselves are not active, but their metabolites, their biotransform products are active. Okay. So we convert inactive drug to its active form or to its active metabolite, which we refer it as a prodrug. So for example, we can take an levodopa. So levodopa is a inactive drug. It does not have any pharmacological action, but when you consume levodopa, the levodopa will be converted into a dopamine, and dopamine will have a very good pharmacological action. So you can see what changes are happening in the chemical structure. So we have to look this from point of medicinal chemistry, and we can easily make out we have removed the carboxylic acid COOH, and we have this NH2 kind of system here. It was here also, but we have only decarboxylated this. Okay. And because of that, the dopamine is formed, and this is active form. This is active form. Okay. So we are converting inactive drug into active form, right? So that is called as activation of inactive drugs. Similarly, we can have an other example of enalapril. So this enalapril is inactive in nature. So this enalapril is converted to enalaprillate, which is active in nature. So this will have a pharmacological action. So you can again make a point of difference here. The OH group is introduced here. The OH group is introduced here, making more polar and active form, which can be easily excreted. So this is a third kind of type or purpose where we activate the inactive drug. They are called as prodrugs. Okay. So now next is the types of drug metabolism. Okay. So basically we have two types of drug metabolism or biotransformation. One type we can refer it as the phase 1. Okay. So phase 1 is also referred as a non-synthetic, and we can also refer it as a functionalization reaction. So what we do in phase 1 or non-synthetic or functionalization reaction, we generally do the generation or exposing the functional group. Okay. So we generate some functional groups, usually the polar functional group, like we can incorporate NH2, we can incorporate SH, OH and so on, so that the molecule will be converted from lipophilic to the hydrophilic, right. Sometimes the drugs undergo phase 1 as well as phase 2 metabolism. So initially it undergoes phase 1 and then it will be undergoing phase 2. So we need to expose the functionalities present on the drug so that it can form a conjugate in the phase 2 kind of metabolizing enzyme, okay, or phase 2 kind of metabolism. So in phase 1, metabolite may be active or it may be inactive. So this is the first type, which involves generally the generation of functionality or exposing those functionality to undergo the conjugation type of reaction, which are mainly happening in the type 2 or phase 2 metabolism. Then second is phase 2, which can also be referred as a synthetic, or it can be referred as a conjugation reaction. So an endogenous radical is conjugated to the drug. So this is the main point of difference between phase 1 and phase 2. In phase 1, what is happening? We are generating the functionalities or we are exposing the functionalities to undergo a conjugation reaction. Okay. And in phase 2, usually what we do, an endogenous radical we conjugate with the drug moiety or the xenobiotic, and these metabolites are generally inactive. There are again several exceptions like glucuronide conjugate of morphine and sulfate conjugate of minoxidil are active. So, these are exception, but generally broadly we can consider the metabolites producing in phase 2, they are inactive, okay, and usually it undergoes the conjugation type of reactions. So, what are the various ways of drug metabolism? Okay. So, what we have understood from the discussion, we are consuming a drug or the xenobiotic, and these drug molecules, after showing its pharmacological action, it should be excreted. Okay. If they are not getting excreted, they will accumulate in the body and they will cause the toxicity. In some cases it needs the activation, and without activation it will not show its pharmacological action, right. So, there are various ways by which the drug metabolism can happen in the body, mainly in the liver. Okay. So, first is some drugs we have discussed, right, it does not require the metabolism. For example, we have seen the chloramphenicol, right. So, they themselves are sufficiently hydrophilic, right. So, they are showing their pharmacological action and they does not require any transformation because they are sufficiently hydrophilic, and that is why they will be easily eliminated from the body. So, this is the first way by which the drugs can be eliminated. So, they usually eliminated in the unchanged form, right. The form of drug what we have consumed, the same chemical form will be eliminated from the body in unchanged form. Okay. So, they does not require the biotransformation, and the number of these kinds of drugs are very small. Second is the drugs which are undergoing the biotransformation or the metabolism by phase 1. So, you can see here the second arrow, the drug has undergone some kind of biotransformation and it has been broken down into some metabolites. So, this is a metabolite of the drug, and this is happening with the generation of the functionality. The drug, lipophilic drug, was converted into a hydrophilic drug which is now getting eliminated. So, this is by phase 1 reaction. Then we have phase 2 reactions. So, in phase 2 we have discussed, usually we form a conjugate reaction. So, conjugation of what? Conjugation of your drug moiety and the endogenous radicals. So, the one which is shown here in the yellow is nothing but the endogenous radical, and this is the drug. So, they are forming an conjugate which we refer it as a conjugate kind of metabolite, and this conjugate metabolite is hydrophilic in nature, and this can be easily excreted in from the body. Okay. And then we have the fourth type which undergo phase 1 as well as phase 2 in a sequential way. So, the drug is converted or it will be broken down or biotransformed into a phase 1 metabolite, and then phase 1 metabolite. So, here we are exposing the functionalities to have a conjugation with that of the endogenous radical. So, this is the drug moiety and the endogenous radical, it has formed a conjugate, and this conjugate will be excreted from the body. So, these are the various ways by which the drug metabolism happens inside the body. Okay. So, in the next session we will see and observe the various reactions of phase 1 and phase 2 metabolism. Okay. So, thanks for watching it.